Okay, we're back again. So we best recap on cell replication as well. In fact, I probably should have included a replication fork in the discussion of Ozaki fragments, but anyway, we can come back on those another time. So remember the cell replication cycle? Hopefully you do, because again, it's stuff you need to know and you may get examined on. In normal cell growth and development, one diploid cell becomes two diploid cells, which are essentially identical. We'll see why that's probably not the case most of the time. And we go through G1, S, G2, and then prophase, metaphase, anaphase, telophase. We do mitosis, and then cytokinesis, and we have two cells. In the gonads, this goes a bit further. And so in the gonads, we produce gametes. So in testes and ovaries, we make egg and sperm. And we then have to do prophase, metaphase, anaphase, telophase a second time. We don't do S, G1, G2 a second time, just mitosis. Well, in this case we call it meiosis. So it's a mitosis a second time. And this now gives us 4 haploid cells. All very easy, isn't it? What could possibly go wrong? Well, plenty. And so we get 2 major changes that can occur in either mitosis 1 or meiosis, so mitosis 2. If it happens in mitosis in your body cell, it generally isn't a problem because the genes are still present and you're not passing it on. If it occurs during the making of gametes in meiosis, then we're looking at potentials for problems, but we're also looking at potential for more variation, more change, more diversity. So again, it can be very useful too, not always a problem. So crossing over and recombination occur during metaphase, and this is because the chromosomes get entangled. And you think about the size of those cells we look at down microscopes, and you realise that in humans there's 2 meters of DNA in every cell that's been chunked up into 46 chromosomes. In metaphase, in mitosis 1, that's 92 chromosomes lined up across the centre of the cell. In mitosis 2, there's only 46 again, but still it's a lot of DNA across a very small space, and so they do get entangled, and that gets recombination of the DNA to occur. The other big one is non-disjunction. So during anaphase, the chromosome pairs split apart and travel to each end, which allows us to produce gametes with a full set of chromosomes, but only one set. And sometimes they don't. Sometimes they stay together and both migrate to the same pole. And this causes a thing called aneuploidy, which we'll talk about now. So, oops, didn't do it again. Come on. Sorry, I think I'm under control now. So aneuploidy is a condition when you end up with 3 copies or no copies of a particular chromosome. In the case of the young Down syndrome person there, we call that trisomy 21. She has 3 21 chromosomes. And interesting enough, that tells a lot about what's on the 21st chromosome, because Down syndrome people are shorter of stature, have poor muscle tone, and have delays in their intellectual development, sometimes quite severe. You can also have polysomy. So, what am I trying to say? This is polysomy, what I'm talking about. So you have more or less than 46 chromosomes. So should be described as having 47 chromosomes. That's not responding again. Come on. Thank you. It's still recording? Yes, good. Oh, playing up. Also polyploidy. And polyploidy is when you get more than one set of chromosomes. So gametes should have one set, they should be haploid. A normal body cell should have 2 sets, it should be diploid. If you get a situation where a human is triploid, so you've got non-disjunction of all of the chromosomes in the creation of a gamete, they tend to get miscarried. You don't get born. So higher evolved animals, you don't see tetraploidy or triploidy occurring. But in some plants, it's not a problem. It often means they're not fertile, but they still live okay. And this also happens often in simple organisms, particularly those things that are capable of asexual reproduction. You get messages from that. So the ability to make gametes, unfortunately, doesn't occur because you've got too many sets of chromosomes. But life can still continue. So polyploidy can be a real issue, but we don't see polyploidy in higher mammals. Okay, so what's all this about? It's all about diversity. So we've now got a whole lot of ways of making variation through mutation, complete and these abnormalities of the chromosomes. And then we've got ways of moving things. So we talk about gene flow. Genetic information flows between populations of particular species. So migration is an important process. It brings fresh genes to a population. And that's one of those concerns we have when a group want to exclude all immigrants. Immigrants bring fresh genes. Fresh genes make diversity. It's really quite simple. So gene flow is just simply about the genes migrating between populations. We also have this thing we call genetic drift. And genetic drift tends to lead, for whatever reason, generally driven by the environment, sometimes culturally as well, to a particular allele being fixed. So one of the interesting examples is when we arrived here in Australia, the Aboriginal population had no B allele for blood. There's only A blood or O blood. You couldn't be AB or A. LB. So the B allele didn't exist. Now, we don't know why that occurred, but that founder population probably didn't bring it with them. Sometimes the environment causes that. Favouring blue eyes and blonde hair would cause that to occur over time. We also have a thing called the founder effect. So that founder effect for Australian Aborigines was quite strong in not bringing the B allele. Martha's Vineyard's another one where the Pilgrims arrived, and the group of people who arrived there had a number of distinct genetic disorders, one of which was deafness. So that deafness, the genetic deafness in this particular population was quite strong for a long time. It's been lots more immigrants into Martha's Vineyard now, but at the time there was a lot of, for a time there was a lot of deafness on this island. It was all from an original member of the first population that arrived there, bringing that with them. So this founder effect is bringing certain conditions with you. We also have a problem with bottlenecks. Further top there is our avifaunal emblem, the helmeted honeyeater, and the cheetah is the often classic example of the founder effect. In fact, humans, we believe, not founder effect, bottleneck. Humans are also probably a bottleneck population. We tend to find only 7 females in our DNA at the start. We can't tell how many males, of course. We'll talk about that later. So this is often a catastrophic event. And for the cheetah, about 10,000 years ago, they were reduced to 1 female with her litter. Now, we can only test females because of their mitochondrial DNA, so we don't have any males who were present at the time. But clearly, the genetic diversity within cheetahs was incredibly low, and it hasn't really built back up that much. Although the population has recovered, the genetic diversity hasn't. We're of time, so we keep moving, haven't I? We talk also about speciation occurring, and the one you need to know for your exam is allopatric, so I'm bolded that, but just because some people are often interested in what other types there are, we talk about allopatric, peripatric, paripatric, and sympatric speciation events. So the diagram there is of a mountain range in Central America, and as that range grew, it would have divided populations of animals and plants on either side, and the environmental conditions on either side will have changed. So over time, with different selection pressures operating, the two populations change to the point that they are no longer the same species. They can't interbreed and produce fertile offspring. And so this mountain range is your classic allopatric situation. So allopatric speciation is about a geographical barrier, a big lake, ocean, river. Peripatric are when an isolated population changes over time with no gene flow. So it's a bit like allopatric, but there's no geographical barrier. So for whatever reason, this particular population has no gene flow into it, and it just changes over time. Paripatric could be caused by extreme changes in the habitat. So the habitat's suddenly changed, and the animals that can survive do, but by doing so, they've created a new species. And sympatric is a process through which new species evolve from a single ancestral species. So you've got one ancestor, and for whatever reason, again, it could be a bit like peripatric, the habitat starts to change, or they've moved into a new area, and certain individuals are favoured. But the only one you need to worry about for the exam is this idea of allopatric speciation. We also talk about reproductive isolation. So there's ways of isolating populations that stop them breeding. And I've got two lovely photographs there. You can't see one for my head, but two beautiful birds there displaying their feathers, the male doing his dance. And so, of course, pre-reproductive barriers mean knowing the dance. It's not just geographical features in the way. You need to know what the dance is to be able to do the mating thing. It can be timing of breeding. There's a wonderful example about mosquitoes on an island, and up the mountain range they breed at different times because of the heat rising through the summer. And so the ones at the bottom of that mountain and the ones at the top of the mountain can't interbreed. They breed at the wrong time. And we also talk about post-reproductive issues. So you can have very high gamete mortality, high zygote mortality, or sterile hybrids. And these things obviously all stop the ability to reproduce successfully. And finally, of course, we talk about selective breeding. A beautiful border collie there. But border collies used for show dogs now, like most show dogs, have so many problems with them because we've bred them in. There's some corridors there in the middle, and a field of corn down below. So most of our breeding selectively has been about producing animals that we could use, or plants that we could use. Now that huge cornfield there is susceptible to disease. It's one corn plant, and it would have very limited variation. And if a disease was to strike that the species had little protection from, it would wipe out at least most of that crop. And the same with the sheep there. If disease hits the flock, the lack of variation within the flock would see it really badly damaged, potentially. So pathogens also show this as they've learnt to get round our antibiotics. We've caused them to selectively breed for resistance to our antibiotics. So not always a good thing. Anyway, that's our first unit. We'll spend some time studying all these ideas and hopefully make some sense of them. And I'm done.